Learning outcomes. After studying this module, you shall be able to know about the toxins of botanical origin, general introduction of certain plant toxins, forensic significance of plant poisoning, and the screening test for the identification of plant toxins. Now let us start with the introduction. The history of poisons and poisoning dates back several thousand years. Early poisons were almost exclusively plant and animal toxins and some minerals. They were used mainly for hunting. Some were used as ordeal poisons, which means ingestion of these substances were believed to be lethal to the guilty and harmless to the innocent. For example, physicostigmine from the Physostigma venenosum, that is Calabar bean, and amygdalin from peach pits. One of the earliest classification of poisons was done by Greek physician Dioscorides, who categorized poisons into three groups, that is animal, vegetable and mineral. An early treatise on plant poisons is De Historia Plantarum by Theophrastus in 370 to 286 BC. The ancient Indian text Rig Veda in 12th century BC also describes several plant poisons. The Greeks used some plant toxins as poisons of execution. Socrates was executed by the administration of hemlock. Now we will study about the forensic issues. Since India is a tropical country, it is host to a rich and varied flora of thousands of plants, and some of which are extremely poisonous. Most people in rural areas depend for their food upon farms and gardens. Cases of accidental poisoning occur not infrequently due to mistaken ingestion of the toxic plant products or the contamination of foodstuffs. Some cases are related to intake of harmful herbal remedies and traditional medicines. A substantial number of cases involve children for whom plants are accessible and attractive. In some Western countries, 5 to 10% of all the human exposures reported to poison control centers involve plants. In India, the overall percentage ranges from 6 to 15%, while if rural populations are taken in isolation, the percentage may be as high as 63%. Now, starting with the classification of plant poisons. The plant poisons are categorized in various classes based upon their action. First, we will study about the neurotic poisons. They chiefly act on the central nervous system or the CNS, such as the spinal cord, and the symptoms usually consist of headache, drowsiness, giddiness, delirium, stupor, coma and convulsions. Examples include Papaver somniferum, Strychnine nux vomica, Cannabis sativa, Erythroxylin coca, Atropa belladonna, Datura festosa, etc. Next are the cardiac ones. These affect the functioning of heart. Example, Nicotiana tabacum, Aconitum, napellus and Digitalis purpurea. Next class is the irritant poisons. These produce symptoms of pain in the abdomen, vomiting and purging. Examples include Abrus precatorius, Calotropis gigantica, Calotropis procera, Cytisus laburnum, Taxus baccata, Croton tiglium, Argemone mexicana and Gloriosa superba. Next are the miscellaneous ones. Cyanogenetic glycosides, ergot, that is Claviceps purpurea, and oleander, that is glycoside. Now we will study about some of the notable plant poisons. So here we will start with strychnine. Strychnine is the primary alkaloid in the strychnos plant, that is the seeds, and is a powerful spinal stimulant. Its botanical name is Strychnus nux vomica. It is also known as dog button, poison nut. This is a tree belonging to the family Loginiaceae, which grows well in South India as well as in certain other parts of the country. The main alkaloid strychnine has been in use as a rodenticide since the 16th century. It is sometimes used for killing stray dogs and hence the name dog buttons. The toxic parts of this tree are the leaves, fruits and seeds. Strychnine and brucine are the principal alkaloids, of which the former is much more powerful. It is a basic alkaloid and can be extracted from the seeds as an odorless, bitter tasting and white crystalline material. The fatal dose is about 30 to 50 milligrams for strychnine and about 1 to 3 grams for the strychnos seeds. Next in line is the poison hemlock. Greeks chiefly employed hemlock for suicidal purposes and as state poison as a form of the capital punishment. Socrates was found guilty for corrupting the youth of Athens with his philosophical teachings and in 420 BC was made to drink the state poison, that is hemlock. Its botanical name is Conium maculatum and hemlock belongs to the family Umbelliferae of genus Cicuta and is a biennial herb that grows erect to an average height of 1 to 3 meters. Several cases of poisoning have occurred and hemlock having been mistaken for parsley, fennel, asparagus and parsnip. The leaves of this plant have a peculiar musky odor which is intensified when they are rubbed in a mortar with some caustic potash. The poisonous properties reside in the piperidine alkaloids, conine and gamma conicine. They are structurally similar to nicotine and possess similar clinical features in toxicity. Next is the Calabar bean. Its botanical name is Physostigma venenosum of the family Leguminosae. This plant is sarcastically used on the west coast of Africa as a test of innocence in cases of suspected witchcraft. The poisonous alkaloid is physicostigmine or acerine. The symptoms of physicostigmine poisoning are vomiting, giddiness, irregular action of the heart. The mental faculties are unaffected. The eyes are bright and pupils contracted, in which latter it differs most strikingly from atropine, hyoscine and daturine, where dilation of the pupil is the rule. Next we will study about Datura. This is a small coarse shrub of Datura species with a strong and rather unpleasant smell, belonging to family Solanaceae, which grows wild all over the Indian countryside. Other common names are Jamestown weed, Jimson weed, thorn apple, stinkweed, devil's weed, angel's trumpet. The toxic principles of Datura are hyoscine, that is the scopolamine, hyoscyamine and traces of atropine, together referred to as belladonna alkaloids. Belladonna alkaloids competitively inhibit the muscarinic effects of acetylcholamine. Sites of action are at the autonomic effectors, inverted by the postganglionic cholinergic nerves, or on the smooth muscles that do not contain cholinergic innervation. Central nervous system effects results from their central anti-muscarine actions, that is vagal stimulation and decrease in heart rate. Usual fatal dose is about 50 to 100 Datura seeds and about 10 to 100 milligrams of atropine, that is usually 60 to 75 milligrams. Next in line is the Digitalis. Digitalis purpurea, the common name of this plant is foxglove, and it grows well in the hilly regions of Darjeeling, Nilgiris and Kashmir. It is a biennial or perennial herb belonging to family Sclerophryaceae, growing up to 1 to 1.5 meters in height. Leaves are hairy, ovate, toothed and grey-green in color, while the flowers are tubular and pink or white in color. There is a related species, that is Digitalis lanata, which is also rich in cardiac glucosides. Leaves constitute the main source of glycosides, which are digoxin and digitoxin, which is the most widely used cardiac glycosides. Other less commonly used glycosides include gitotoxin, gitalin, digitonin, digitin, lanatoside C and deslonaside. Next, we will study about Argemone mexicana. Other common names of this type are the prickly poppy, yellow poppy, Mexican poppy. This is a robust, prickly, annual or perennial herb belonging to family Papaveraceae, which grows up to 4 feet in height, bearing thistle-like leaves and yellowish flowers. There is no legitimate use for the argemone seeds or the oil that is extracted from them. In India, mustard oil and other vegetable oils are often adulterated deliberately with argemone oil. Sometimes the dark variety of mustard seeds is adulterated with argemone seeds. The toxic part, that is the seed and expressed oil, are quite toxic. Leaves are also toxic to a lesser degree, and the toxic principles of argemone are sanguinarine and dihydrosanguinarine. Both of them are physiologically active benzophenthridine alkaloids. In addition, the other alkaloids of lesser importance are present, such as protropin, berberine, clerthrine, isoquinoline and eosin. Berberine and protropin are found throughout the entire plant, while sanguinarine and dihydrosanguinarine are found in the seeds. Liver, heart, kidneys and lungs are the target organs of argemone alkaloids, and it is postulated that membrane destruction is the probable mode of action. The exact mechanism is not well understood. Next is croton. Croton tiglium, belonging to the family Euphorbiaceae, grows well in Assam, Bengal and the Western Ghats. It is a small evergreen tree with ovate or elliptical leaves, which are narrow pointed, toothed and 2 to 4 inches long, varying in color from metallic green to bronze, orange or yellowish. The seeds, oil and root extract are used as a drastic purgative in traditional medicine. The toxic principles of croton are croton or toxalbumin and crotonicide, that is glycoside. Next, we will study about ergot. Ergot is produced by a fungus that is Claviceps purpurea, which infest certain types of grains, especially rye. The spores of the fungus are carried by insects or wind to young rye, where they germinate into hyphae or the filaments. The hyphae penetrate deep into the grain and harden into a purplish structure called sclerotia, which particularizes a number of the ergot alkaloids. During wet seasons, Claviceps purpurea can infest wheat, barley, rye, which is the most common, oats, wheatgrass, quackgrass, smooth brown grass, wild rye and blue grasses. Examples of ergot alkaloids include dihydroergocornene, dihydroergocristine, dihydroergocin, dihydroergotamine, dihydroergotaxamine, ergobacin, ergocornin, ergocristin, ergocryptin, ergocin, ergometrin or ergonovine, ergotamine, ergotaxamine, methyl ergonorine, bromocriptin, lergotril, lyseride, lysergol, metergyline, methyl ergovanine and methyl sergide. All these are the derivatives of 6-methyl ergoline, a tetracyclic compound. There have been more than 350 chemicals identified, but less than 10 are used therapeutically. Natural ergot is also the source of potent hallucinogen lysergic acid diethylamide or LSD. Next is aconite. This plant belongs to family Ranunculaceae and grows well in the hilly regions of northern and eastern parts of India, extending from Assam to Kashmir. The tuberous part of this plant is very popular in Chinese medicine for the treatment of various ailments. The root is usually processed by drying, soaking or boiling, which significantly reduces its toxicity. Raw aconite roots are highly toxic. Herbal decoctions of aconite are generally prepared by soaking the roots in water or saturated lime water and then by boiling. This causes hydrolysis of aconite alkaloids to less toxic benzyl aconine and aconine derivatives. Aconite is also used in Indian folk remedies. It is also used as an antipyretic in Ayurvedic medicine and after detoxification. Aconite is sometimes used as an abortifacient. Fatal dose of aconite is about 9 to 18 grams of root and about 3 to 5 milligrams of aconitine, while about 10 to 15 milliliters of tincture. However, deaths have been reported with as little as 1 gram of root, 0.2 milligram of aconitine and 5 ml of aconite tincture. It has been estimated that an adult lethal dose is generally about 1 grams of plant part, that is root, 5 ml of the prepared tincture or 2 milligrams of pure aconite. Next is the extraction of plant poisons. Extraction is important for the analysis as a detection and identification purpose. Different methods which are classical and instrumental in nature are solely dependent on the efficiency of extraction, which is dependent on a number of factors which are analytical requirements, nature of sample and the nature of poison, whether the alkaloid or glycoside it is. The plant materials like root, stem, leaves and seeds, that is the non-biological matrices, are required to be extracted from. In this table we have on one column the plant poisons and on second column we have shown the fractions in Stas Otto method. Starting with the Papaver somniferum, the fraction obtained is basic chloroform extract. Next is Strychnine nux vomica. For the fraction obtained is basic chloroform extract. For Cannabis sativa it is acetic ether extract, and for erythroxylin coca it is the basic chloroform extract. For Atropa belladonna it is basic chloroform extract. Next is Datura fustosa, basic chloroform extract. For Aconitum napellus it is the basic chloroform extract. For Digitalis purpurea it is acidic ether extract. For Abrus precatorius the fraction obtained is of acidic ether extract. For Calotropis gingentia or procera it is acidic ether extract. For Citius laburnum it is basic chloroform extract. Next is Taxus baccata, basic chloroform extract. Next is Croton tiglium, it is the acidic ether or the basic chloroform extract. For Claviceps purpurea it is alkaline ether chloroform in the ratio of 3:1. Extract has to be obtained for the Stas Otto method, and for oleander it is acidic ether extract. Next we will study about the presumptive tests of the active elements of plant toxins. The purified extracts of samples of plant materials or other materials, that is the biological or the non-biological, are subjected to analysis by various color tests for their preliminary identification. Starting with the test for opium alkaloids, first we have the Marquis test. To the dried residue of extract in a porcelain basin, that is the flat type, 1 drop of Marquis reagent is added and a purple red color is produced, which changes to violet and finally to blue. Next test is of Huysmans test. To a little dried extract in a porcelain basin, 2 to 3 drops of concentrated sulfuric acid is added and heated on a water bath for 30 minutes over a small flame for a few minutes until white fumes appear. A reddish or reddish brown or black color appears. It is cooled and 1 drop of concentrated nitric acid and a crystal of potassium nitrate are added. A reddish violet color appears, which immediately changes to blood and then to reddish yellow and finally fades away. Next is Fraud's test. To the dried residue of extract, a few drops of Fraud's reagent are added. A violet color changing to green and finally blue is observed. Next is the urotropin test. To a little dried extract in a porcelain basin, a few drops of the aqueous solution of urotropin is added and warmed slightly. A purple color changing to blue and then green is observed. Next is the test for porphyroxine. 2 drops of acetic acid solution of the residue of extract is taken in a porcelain basin, followed by the addition of a few drops of dilute hydrochloric acid. It is warmed over for a low flame and a pink or rose red color is produced. Next comes the test for meconic acid. A few drops of neutral solution of the ferric chloride are added to 1 to 2 ml of an aqueous extract or the extract that has been obtained after separation of the meconic acid. A blood red color is produced which is not destroyed by boiling or by adding hydrochloric acid or mercury chloride solution. Next is tests for strychnine, starting with first of all the Mandelin's test. First of all, it is necessary to separate strychnine from brucine. The chloroform extract, that is the basic fraction containing strychnine and brucine, is evaporated to dryness. 2 ml of dilute sulfuric acid is added, followed by 2 drops of concentrated nitric acid. It is allowed to stand for 30 to 60 minutes at 10 to 20 degrees Celsius and thereafter made alkaline with sodium hydroxide solution and extracted several times with chloroform. The chloroform extract is combined, washed and evaporated to dryness. The residue does not contain brucine. To the brucine-free residue, add 1 drop of Mandel's reagent, that is 1% solution of ammonium vanadate in concentrated sulfuric acid, and a deep violet blue or deep purple color appears which finally changes to yellow on long standing. Next is the play of colors test. To the brucine free residue of extract in a porcelain dish, 1 drop of pure concentrated sulfuric acid is added. No coloration is observed. A crystal of potassium dichromate is drawn by a glass rod through the sulfuric acid. A play of colors is observed. First, a momentary blue changing to a violet color, which gradually changes to reddish purple, red or orange, and finally to yellow. Now, test for brucine. Starting with the nitric acid test. To the dried residue of the extract, 1 drop of concentrated nitric acid is added. A blood red color appears which changes to reddish yellow and finally to pure yellow. To this, a few drops of stannous chloride solution is added and an intense purple color develops which is destroyed by adding a few drops of concentrated HNO3. Next is Vitali's test. To a little dried residue of the extract in a porcelain dish, 1 to 2 drops of fuming or concentrated nitric acid is added. It is evaporated to dryness on a water bath. It is allowed to cool and a few drops of freshly prepared alcoholic caustic potash solution is added. A fine violet color is produced which changes to orange red or red and then disappears. On adding a few drops of excess alcoholic caustic potash solution, the color reappears. Next, we will study about the test for cannabis sativa, starting with fast blue bee test. A small amount of residue of the extract is placed in a test tube and a very small amount of solid fast blue bee reagent, that is the solid fast blue bee, is to anhydrous sodium sulfate in the ratio of 2.5:100 is added. 1 ml of chloroform is then added and shaken. It is kept for 2 minutes and the chloroform layer becomes purple red in color. Next is the Duquesnoy's Levine test. A small amount of residue of the extract is placed in a test tube. It is shaken with 2 ml of a reagent containing acetaldehyde and vanillin, that is 5 drops of acetaldehyde and 0.4 grams of vanillin dissolved in 20 milliliters of 95% ethanol for 1 minute, and in 2 ml of concentrated hydrochloric acid is added, shaken and allowed to stand for 10 minutes. If a color develops, 2 milliliters of chloroform is added. the lower chloroform layer becomes violet. Next is the test for cocaine. Starting with first is the Scott test. The residue of the extract is taken in a test tube and 5 drops of 2% cobalt thiocyanate solution, that is prepared by dissolving 2 grams of cobalt thiocyanate in water and diluted with 96% glycerine in the proportion of water to glycerine, that is 1:1. The mixture is then shaken and a blue color develops at once, which indicates the presence of cocaine. Methaqualone also gives the positive reaction. If a blue color develops, 1 drop of concentrated hydrochloric acid is added, the blue color disappears and a clear pink color appears. If a blue color does not disappear, 1 drop of concentrated hydrochloric acid is added. Then a few drops of chloroform are added and shaken. The chloroform layer becomes intense blue. Second is the gold chloride test. In this, 1 drop of 5% gold chloride solution in water is added to a few drops of the extract. A precipitate is formed which appears as delicate rosette or long rod-shaped crystals under microscope. The precipitate is also insoluble in dilute hydrochloric acid. Next are the test for Atropa belladonna. Starting with this is the Vitali's test. In Vitali's test, a portion of residue of the extract is treated with a few drops of fuming nitric acid in a porcelain basin. It is evaporated to dryness on a water bath. The residue is then cooled and moistened with a few drops of freshly prepared alcoholic caustic potash solution when a violet color is produced, which soon changes to red and finally disappears. The color may be made to reappear by adding more alcoholic caustic potash solution. Next is the Gerard's test. In this, 1 to 2 ml of 2% mercury chloride solution in 50% of alcohol is added to a portion of residue of the extract. A red color develops immediately. Hyoscyamine produces a yellow color which becomes red on burning, while hyoscine does not produce any change of color. Next is the test for Datura fustuosa. Starting with this is the Vitali's test. In this, a portion of residue of the extract is treated as I have already said above, and when violet color is produced, which immediately changes to red and then disappears. On adding a few drops of alcoholic KOH, the color reappears. Next is the test for Nicotiana tabacum. Starting with this is the Mayer's test. Potassium mercury iodide prepared by dissolving 1.357 grams of mercury chloride and 5 grams of potassium iodide in 100 ml of water. The dried residue of extract is acidified with acetic acid followed by the addition of 2 drops of reagent. A white or yellowish precipitate is obtained. Next is Phosphomolybdic acid. To the dried residue of the extract, a few drops of phosphomolybdic acid is added and warmed. A yellowish white PPT is obtained. Next is the silicotungstic acid. To the dried residue of the extract, a few drops of silicotungstic acid are added. A white PPT is obtained. Next, we'll study about the test for Aconitum napellus. First is the Pallet's reaction. To the purified extract of the residue, a few drops of a mixture containing 2.5 grams of syrupy phosphoric acid and 0.1 gram of the sodium molybdate is added and heated over a small flame until vapors appear. A violet color develops. Next is the Alvarez reaction. To the purified residue in a porcelain dish, 5 to 10 drops of pure bromine is added and evaporated to dryness on a water bath. 1 to 2 ml of concentrated nitric acid is added and evaporated to dryness and a few drops of bromine are to be added if nitric acid loses its color. To the yellow oxidation product, 1 ml of the saturated alcoholic solution of sodium hydroxide is added and again evaporated to dryness. A red or brown residue is obtained. It is allowed to cool and 5 to 6 drops of a 10% copper sulfate solution is then added. A green color develops. Next is the tests for Digitalis purpurea. The dried residue of the extract is taken in a small porcelain basin, and to this add 5 drops of 1:1 mixture of ethanol and chloroform is added and stirred. The organic solvent extract is spotted on a number 1 filter paper in varying amounts and is sprayed with a 10% solution of antimony pentachloride in chloroform. The development of yellow color changing to purple is observed. On warming the spotted paper in hot air for 5 minutes, the color of the spot changes to black. Next are the tests for Abrus precatorius. Starting with the first is the fast blue B potassium hydroxide test. In this, to the dried residue of the extract in a porcelain basin, a few drops of 5% ethanolic solution of fast blue B salt is added, followed by 2 drops of aqueous KOH solution. A red to orange color is observed. Next is the Marquis test. To the dried residue of extract, 2 drops of Marquis reagent, that is prepared by mixing 1 volume of formalin or formaldehyde solution with 9 volumes of concentrated sulfuric acid is added. A pink color is formed. Third is the Van Eyck reagent. To the dried residue of the extract, 1 drop of Van Eyck reagent, that is prepared by dissolving 1 gram of para-aminobenzaldehyde in 100 milliliters of ethanol and by adding 10 ml of hydrochloric acid is prepared. A green color changing to blue is observed. Next is the special test or the agglutination test. To 2 drops of the aqueous solution of residue of the extract is added 2 ml of defibrinated blood, that is undiluted, in a small test tube. The red blood corpuscles agglutinate into a mass like that of a sealing wax. Next comes the color test for Calotropis species. First is the concentrated hydrochloric acid. To a small portion of the residue of extract, a few drops of concentrated hydrochloric acid is added and slightly warmed. A greenish blue color is formed. Next is the concentrated sulfuric acid test. To a small portion of residue of the extract, a few drops of concentrated sulfuric acid are added. A pink to purple color develops after a few minutes. Third is the Frod's reagent test. To a small portion of residue of the extract, 2 drops of Frod's reagent is added. A deep green color changing to blue and finally to green color develops. Next, we will study about the color test for Taxus baccata. Starting with the first, we have concentrated sulfuric acid. To the residue of extract, 1 drop of concentrated sulfuric acid is added. A violet color is produced which disappears on addition of water. Second is the nitric acid. The solution of residue of extract is prepared in sulfuric acid using a few drops of acid. To the acid solution in a small quantity, 1 drop of concentrated nitric acid is added and a rose red color is formed. Next is the molybdic acid in sulfuric acid test. To the residue of extract, 2 drops of molybdic acid in concentrated sulfuric acid is added and a rose red color is produced. Now we will study about the test for Croton tiglium. Starting with test 1, we have 2 ml of concentrated extract of residue in ethanol is added to an equal volume of 40% sodium hydroxide solution in a small test tube. A brownish red or reddish violet ring is developed at the junction of 2 liquids. The color formation is rapid by warming process. Next is test 2. 2 ml of the concentrated ether extract of residue is taken in a porcelain basin and the solvent is evaporated off. To the residue and alcoholic solution of 1% solution of para-dimethylaminobenzylaldehyde in rectified spirit is acidified with 1 ml of concentrated sulfuric acid added drop by drop. A transient red color is observed. On evaporating to dryness on hot water bath, the residue becomes brownish red to purple in color, which changes to pale blue on adding of the excess of reagent. Next comes the test for Argemone mexicana. Starting with the first test, we have nitric acid test. A few drops of oil is mixed with an equal volume of concentrated nitric acid when a crimson orange color appears. Second is the cupric acetate. When 1 ml of glacial acetic acid and 2 ml of cupric acetate solution are added to 5 ml of the oil sample in a test tube and then boiled on a water bath for 15 minutes, greenish discoloration occurs. Next test is for ferric chloride. 2 ml of concentrated hydrochloric acid is added to 4 ml of the oil, mixed thoroughly and warmed on a boiling water bath for 4 to 5 minutes. 1 ml of ferric chloride solution that is prepared by dissolving 10 grams of fresh ferric chloride in 10 milliliters of concentrated hydrochloric acid and making up the volume to 100 ml by distilled water is added and again heated on a water bath for 10 minutes. A precipitate of reddish brown color appears. The precipitate is acicular or needle shaped when observed under the microscope. Next comes the test for ergot, starting with the Marquis reagent. To a portion of the dried residue of the extract, 1 drop of reagent is added and a brown color develops. Second test is the Vitali's test. The procedure for the test have also been stated earlier, as I have already stated in this module. A play of colors from dull orange to yellow and then purple is observed with the residue of the extract. Third is the Mandelin's reagent test. To a portion of the dried residue of the extract, 1 drop of reagent is added. Purple brown color develops. And lastly, the Frohd's reagent. To a portion of the dried residue of extract, 1 ml of reagent prepared by dissolving 0.5 grams of ammonium molybdate in 100 ml of water is added. A color change from deep green to red, grey and finally blue is observed. Now, I will summarize all that we have studied in this module. Phytotoxicology is an expression used to signify the study of plants that produce or evoke explicit deleterious effects on human. Poisons originating from plant are known as plant poisons. The toxins may be present in any part of the plant like leaves, stems, seeds, roots, barks, etc. Plant poisoning is more accidental in nature due to the unintentional consumption of poisonous berries, fruits or plant bodies, but homicidal activities may also be encountered many instances. The ancient Indian text Rig Veda in the 12th century BC also describes several plant poisons. The Greeks used some plant toxins as poisons of execution. Socrates was executed by the administration of hemlock. De Historia Plantarum by Theophrastus contains the description of plants having poisonous characteristics. The Abrus papyrus circa 1500 BC contains information pertaining to many recognized poisons including hemlock, the state poison of the Greeks, aconite, a Chinese arrow poison, and opium, used as both a poison and an antidote. The plant kingdom contains potentially 3 lakh species, and the toxic effects of plants serve primarily as a defense mechanism against the natural predators. Toxicity in humans can result from simply touching as well as ingesting plants to cause a truly wide array of deleterious effects. The toxic effects on humans can range from simple hay fever caused by the exposure of plant pollen all the way to serious systemic reactions caused by the ingestion of specific plants. Thank you.